Hello, everybody. Welcome to VanBug, our February session for year 2022. I'm excited for today's talks by both our student talk, introductory student talk, and our main keynote speaker. So to start off, as usual, just a quick schedule. We're running 5 minutes off the schedule already. So I'm just going to have quick announcements. And then we're going to have our student talk and then our main feature talk, with potentially staying until up to 1 if you have any Q&As or want to discuss or want to socialize. No commitment for that part, for the later part there. And in terms of questions for the speakers, speakers are free to take questions throughout their talk if they wish to, or if you wish, you can take the questions at the end. Me and like the other hosts will be moderating the questions, either reading them or asking the questioner to speak up and say their question. So with that, want to thank our sponsors. So VanBug is sponsored by the Graduate Studies program in Bioinformatics at UBC and SFU. We are also sponsored by Genome BC, by Ecoscope program at UBC, and by the Bioinformatics, Canadian Bioinformatics workshops, and Vancouver's local Abcelera, and Langara College here in Vancouver. And we are also sponsored by Stem Cell Technologies. And we are also affiliated by 2 other sister bugs, Montreal and Toronto, Monbug and Torbug. And we are part of the International Society for Computational Biology as a regional group. So with that, I would like to introduce our speakers for today. Very exciting 2 talks. First, we have Armita Safa from the Faraz Haj's lab at Vancouver Prostate Center and UBC. And she will be introducing her talk on discovery of mid-range novel sequence insertions using long-read sequencing. I'm excited to see this presentation because I'm a bit biased. Armita is in the same lab as me, so I think it's going to be a nice talk. And then for our main speaker, we have Dr. Sandrine Moreira, and this is going to be a very thrilling talk. So Dr. Sandrine is an associate professor in the Department of Biochemistry and Molecular Medicine at the University of Montreal. And she leads the SARS-CoV-2 genomic sequencing efforts at the Public Health Laboratory of Quebec. And before that, Dr. Sandrine received her PhD in bioinformatics from the University of Montreal in 2016, with a focus on RNA editing and trans-splicing of mitochondrial genes of a marine microbiome, a marine microbe. And she's worked at the Pasteur Institute of Paris, and she developed bioinformatics tools for microbial genomics. Her interests are in genomic research, and with a special focus on translating such research projects from basically discoveries and fundamental cutting-edge discoveries to clinically useful tests. More recently, and the topic of her talk is her key role in developing the genomics program for the SARS-CoV-2 surveillance at the Public Health Laboratory of Quebec. She is also the co-PI in a large Canadian COVID-19 genomics network and participates in the Canadian Network for Modeling Infectious Diseases. So with these introductions, I'll start with Armita. Please go ahead for your talk. And do you prefer to take questions throughout, or do you want to keep them to the end?
I think end would be better because the flow of the algorithm. Let's go.
Okay. Okay. Yeah. Cool. So I'll give you the floor, and please share your screen.
You can see my screen now?
Yes.
Okay, great. Well, thank you, Bara. Hello and good morning, everyone. I am Armita Safa, a master's student in bioinformatics at UBC under the supervision of Dr. Faraz Haj at Vancouver Prostate Center. And today I will be presenting my project Rinsler, which is a tool for the detection of mid-range insertions using long reads. First, a bit of background. Genomic variations are defined as differences in the DNA of individual members of the species. They can provide insights into evolutionary processes that have shaped genetic diversity, as well as differences in susceptibility to diseases. Some of these variations might also be harboring coding exons and regulatory elements. Genomic variations might be in the form of single nucleotide variations, which is an alteration in one single nucleotide, or they may be in or they may be bigger alterations, which are called the structural variants, and they are in different forms like deletions and insertions. SVs constitute a substantial proportion of genomic differences, but have been understudied because they are more difficult to identify. There are several tools for detecting SVs, many of which are based on short reads. In principle, taken individually, each type of SV induces a different pattern in mapping reads that can be used to infer the variant. For example, a deletion forms a lack of sequence, and therefore you can see a gap in the alignment. In this project, we have focused on one class of SVs called novel sequence insertions. This class is harder to detect because there is a missing sequence that needs to be reconstructed and thus requires assembly. Current discovery tools use a similar strategy to each other, where read pairs that do not map to the reference genome, called orphans, are flagged as possibly originating from an insertion sequence, and one end anchored read pairs, where one end maps to the reference and the other end does not, likely point to the insertion breakpoint. Now, this brings us to our problem. Characterization of novel sequence insertions is still lagging, especially in insertions longer than read lengths, primarily because insertion sequences can only be discovered through assembly, which can be computationally challenging and also may lead to incorrect or fragmented sequence reconstructions, for example, due to repeats. One approach to overcome these issues is using long reads, but there is a caveat that long reads are more error-prone. Taking the strengths of both short and long reads into consideration, Rinsler takes a hybrid approach in which short reads will help us identify breakpoint locations with high accuracy, and long reads will aid us in assembly by building more contiguous sequences and also resolving repeats better. Here you can see the overall workflow of Rinsler. The input to the workflow is a mapping of the short reads and their matched long reads. And I'll go into the details of each step in the following slides. The first step is pre-processing, which itself consists of two substages. First, Rinsler scans the short read alignment file and looks for signals that might point to a potential insertion breakpoint. For example, the reads in that region will partially map to the reference, but the rest of the read will be seen as a soft clipping. And if a certain number of reads are all in favor of a location, we mark this location as a breakpoint, create a cluster, and add all those short reads that were supporting it to the cluster. The next step is creating a fingerprint index of the long reads. Fingerprint indexing is the process of generating a summary of the data to be able to rapidly query it and get approximate answers. It's like summarizing a text to its keywords and then searching by keywords. We do this by sliding a window on each read, and in each window, we pick the k-mer that has the lowest hash value, called the minimizer, and that would be the representative of that window. For example, here, the green diamond is the minimizer of the first two windows. And so we add the other minimizers as well, and continuing this procedure, we end up with the full index that has all the minimizers and which reads and where in the reads they are found in. Then we proceed to long read identification, which itself consists of three steps. So far, we have a set of breakpoints and a fingerprint index from the long reads that we can query. Now, our goal is to find the long reads that are covering the region around the breakpoint. To do this, we extract a genomic anchor, for example, on the left side of the breakpoint, and then use it as the query to our fingerprint index. This will provide us with a set of long reads that potentially map to this region. Now, if we have short read content from the insertion, we can further filter these long reads and keep those that are covering the region as well as going into the insertions. And we repeat this procedure for the genomic anchor on the right side. So at the end of this stage, we have two sets of candidates, left anchored and right anchored. These candidates all contain some percentage of the minimizers from the genomic anchors. But since we only use the summary, we don't know if these minimizers are happening in order and whether they are actually providing a good mapping. So we do a fast approximate local chaining from the long reads to the anchors. Here, R1 is an example of a good chain. The order of the minimizers is preserved on both chains, and the distance of the beginning and the end of the chains are approximately similar. However, for R2, as you can see, they're not in the same order on the genome, so we discard that one. For good candidates, we identify an interval based on the location of the chain that corresponds to the part of the read that is mapping to the anchor and breakpoint, because the rest of the read is not in our region of interest, so we don't need it. So now we have left anchored reads, right anchored reads, and we have the intervals on them. First, we clean them up by trimming the sites. And now, when we look at them, we see that some reads, like R6 or R7, are in both sets. There are two cases that can cause a read to be picked from both sides of the breakpoint. Either the read is reference allele supporting read, so it contains both anchors, but it does not contain the insertion. Or the read contains the insertion as well as the anchors, which we call these reads bimodal reads. We differentiate between these two cases by looking at the chaining information. In a reference allele supporting read, we expect the mapping location of the chains to be on the right and the left, to be within close vicinity of each other, as is the case for R7. However, in R6, we see that the chains have quite some bit of distance, so we can call this a bimodal read. So we detect the bimodals and discard the reference supporting reads. So now we have our trimmed and classified reads, and we need to move to sequence construction. Depending on the sets of candidates that we have, there are two possible scenarios. Case A is that we have left anchored, right anchored, and bimodal reads. So we can reconstruct the whole insertion. We use SPOA to create a multiple sequence alignment of the reads, and then based on the MSA and the profile, we build a high quality consensus. Case B is that we don't have any bimodal reads. Hence, we can only partially build the sides of the insertion, but not the middle part. What we do here is that we feed those partially built sequences as anchors to the identification module that we saw earlier, and we get a new set of reads that is now further into the insertion. This way, we gradually build a bridge from both sides of the insertion to each other. Like to visualize this, the green and orange segments are the partially built sequences from the first stage. And using them, we can pick new reads and extend our partial sequence. And we repeat this procedure until we pick bimodal reads, because that means that the bridge is completed from the sites. And now for identifying the insertion itself, we do a semi-global alignment from the constructed sequence to the genomic region around the breakpoint. And this way we can discover the exact breakpoint location along with the insertion content. And we can also move on to genotyping the event. We simulated a data set for testing and parameter tuning purposes. Sequences from Methylobacterium genome were inserted into chromosome 21 of human genome, and using simulators, we generated long reads and short reads. For an event to be correct, we expect it to be within 20 base pairs of the actual breakpoint in the ground truth, and to match the actual insertion sequence by at least 90% similarity. What we saw was that Rinsler has a consistently high recall and precision. And also we tried subsampling the long reads to 20x coverage. And as you can see, there's only a negligible drop in recall and precision values. We can also see that compared to PAMIR, which is a short read only tool, Rinsler performs better in terms of recall and precision, and the difference is especially more significant in longer insertions, which is where short reads lag the most in the assembly step. So with that, I would like to thank my supervisor, Dr. Hatch, and all my colleagues at Hatch Lab, and also our collaborator, Dr. John Alcon, for their help steering this project. Thank you, and I'll be happy to take questions now.
Okay, so thank you so much, Armita. If anybody has a question, please, you can either write it up in the chat or you can use the raise hand function and you can speak your question. I guess I can ask a question about, so in the simulation experiment, the inserted sequences are from a completely different genome, which is the, I guess it was a bacterium. But do you think that if the inserted sequences are not novel, that you might mistake them as novel sequences if they're like translocations, or maybe like something that is happening there, but it's actually similar in sequence to some other part of the human genome? How would that affect your work?
Well, that is certainly a challenge to detect which ones are novel and which ones are not. Right now, we try to use different repeat events that we already know about to check for the, to try to discard all of the repeat events. So only we have like what are called non-repeat insertions. But that is always a problem because you can't like say 100%, we may still miss some of them as that are repeats, but we detect them as novel. But the thing is that because we are using long reads and we are building longer segments, we expect this problem to be reduced because we want to resolve the repeat regions better.
Mm-hmm. Thank you. Well, we have a question from William. Do you have plans for comparing Rinsler to other assembly algorithms designed for long read sequencing data? So just doing full-on assembly, I guess, and how does that compare to doing something that is specific for novel insertions?
Well, Rinsler is assembly free, so we only do the assemblies in the regions that we are interested in and from longer segments. So we don't do an end-to-end assembly of the genome. And as far as I know, we have long read tools that are used for structural variant detection, but they all use mapping data and not this algorithm that we have proposed here. But comparing to assembly algorithms, if by assembly you mean an end-to-end assembly and then aligning it to the thing, there are some tools that we are comparing against, but the thing is that even with those, because we won't be able to get a perfect end-to-end assembly, still the events that are larger and longer would be missed, as we can see like right now when comparing to tools such as Sniffles or SPEM, we can see that they are missing longer events that we're not.
Mm. Do you think it's, so if I followed like fully like correctly, you're not using the short reads in the assembly step for Rinsler, right?
No.
Do you think there is use for them in the assembly step? Because you use them just to find like anchors and breakpoints.
Yeah. Short reads are mainly used for locating the breakpoints, and then we can use them to filter out some of the long reads, because if the short reads are aligning to somewhere or like one ends mapping, we know that the part that is not mapping belongs to the insertion. So we can use that part of the short reads to discard the long reads that are reference allele supporting or very high error, so they don't help us. But that is the use that we're getting out of short reads, filtering and locating breakpoints. We don't actually use them in assembly.
Okay, cool. Well, I think that's all the time we have for your talk and the Q&A. Thank you so much, Armita. And with that, I'll hand it over to Dr. Sandrine. Please share your screen and start your presentation. And do you prefer to take questions throughout as you go, or do you want to keep the Q&A for the end?
Well, I can take questions during my talk because there are different parts, so maybe it would make more sense. But what people want, that's okay.
Okay, so if anybody in the audience have a question, please write it in the chat, and then me or one of the hosts can see like an opportune moment and like maybe speak up and ask it for you.
Okay, and this is it. Okay.
Okay, perfect.
Okay. So thank you for the invitation and this opportunity to present what we are doing in Quebec for SARS-CoV-2 surveillance. So you already did a nice introduction, thank you. So I'm the lead genomics and bioinformatics in the Public Health Lab of Quebec. So you will see the LSPQ, Laboratoire de Santé Publique du Québec, in my talk. So this is the French word for Public Health Lab of Quebec. And this is my mail if you want to send me a question after the talk. So the outline of my talk, so of course, as I told Bara at the beginning of the talk, I have too much material, but that's okay, so I can stop earlier. So I will start with the most important aspect, and then if we have time, I will go through the other parts. So the first point will be the SARS-CoV-2, the description of the SARS-CoV-2 surveillance network in Quebec. And I will highlight the successes and also the challenges of this surveillance program. And then I will describe some investigations that we did. So I will walk you through an outbreak investigation, how we process for outbreak investigation, and I will compare two investigations in long-term care facilities. And in the second part, I will describe the early introduction of SARS-CoV-2 in Quebec. So a great work that we have been doing in collaboration with the team of Jessie Shapiro. And the third part would be about neutralization assays of Omicron, if we have time, and also some thoughts about the genomics in public health laboratories and the lessons learned, and my thoughts about the future of genomics in public health labs. So the surveillance network in Quebec, so of course, everything started almost 2 years ago. And I remember in the lab when we heard about this new virus, the first thing that I did is that I walk right into the office of my boss, the director of the Public Health Lab, and I said, well, we have to sequence those viruses. And he said, well, yes, but that's not the priority. We need to develop a diagnostic test first. Sequencing is really not a priority. Okay, and then 1 week after, same thing, we have to sequence the virus. And every week after, I went to his office saying that we have to sequence the virus. And after that, he was so annoyed, I would say, that he said, well, okay, do whatever you want, you have my blessing, but I tell you, I have no resources for you, no money, so you do whatever you want, but I have nothing. I was like, okay, no resources, no money, that sounds like an academic research program. And okay, so I talked to everybody I could find about sequencing sequencing SARS-CoV-2 at the beginning, and I mean, we were very lucky in Canada. So I talked to every sequencing centers in Quebec, and I had extremely good contact with those partners, which are now a partner in our surveillance network. But we were very lucky also to have in Canada this fantastic program, which is the Canadian COVID Genomic Network. And it was a program which was set up at the beginning of the pandemic, and I think that other people in Canada were just like me, saying we have to sequence SARS-CoV-2, we have to, and finally I guess that people were convinced, and we had this huge 40 million project with the goal of sequencing 150,000 viruses at the beginning. So we have sequenced right now far much viruses and 10,000 hosts. It's a pan-Canadian network with Public Health Lab of provinces and territories, with the National Microbiology Lab, sequencing centers, also hospital, researcher, and industry. And among these $40 million project, so we had almost 10% for the Quebec. And you can learn more about that program on that link. And 1 year after the beginning of that project, it was at the beginning of 2020, finally, Quebec realized that it would be interesting to put money into the sequencing of SARS-CoV-2, and they added a $7 million project for specifically the surveillance of SARS-CoV-2 variant. So remember, it was beginning 2021, so it was the alpha variant. So it was just the beginning of the variant thing. And so it was clear for everybody at that time that we had to do surveillance of the population of variants which are circulating in Canada. And the goal for Quebec was to sequence 65,000 viruses in 2021. And we are very happy because that program was renewed 2 weeks ago. So we will have funds for sequencing for 2022, which is good. So the SARS-CoV-2 surveillance network in Quebec. So this is a project that we call CAFseek, and there is a website, not that much information, but still you can have a look. So we have different components in that network. So first the hospitals. So the hospitals collect the clinical specimen, they do the testing and sometimes the screening, depending on if we do screening or not for variant. And something in Quebec, not specific, but which is difficult, is that we have most than 100 testing sites in Quebec. So 100 sites that will send their clinical specimen eventually for sequencing. And so we have set up this biobank that we call CAFbank, which stores, as for now, more than 370,000 COVID samples. So it's samples since the beginning of the epidemic. So it's a fantastic resource for research, and I hope that this resource will be open to researchers. So as for now, it's only for public health and a collaboration with public health in Quebec. So the hospitals, they collect the samples, they send to the biobank, and then the biobank do the aliquoting and sends the aliquot to sequencing sites. So there are 3 sequencing sites in Quebec, so LSPQ, so my lab, McGill Sequencing Center, and Genome Quebec Sequencing Center. And I can't say enough how incredible were those persons in McGill Sequencing Center and Genome Quebec to support the project since the beginning of the pandemic. It was really, it's really fantastic what they did. So more than 70,000 samples have been sequenced, and that represents 15% of COVID positive tests. I should say that it represented 50% of COVID positive cases, because with the Omicron, the huge Omicron wave, it's more like 8% of COVID positive cases now. So we have also collaboration with researchers, academic partners, and of course link with public health authorities and ministry, and link with epidemiologists, which is very crucial for the collecting the information about the patients. Our role in that surveillance network is to develop and validate methods, so the role of the public health lab. We have an ISO certification, so we can provide scientific support and materials for certification, so panels, documentation, etc. We provide guidance and recommendation for SARS-CoV-2 screening and sequencing. We coordinate the sequencing in Quebec, and we have strong connection with epidemiologists as part of the INSPQ. INSPQ is a National Institute for Public Health in Quebec. So I jump into the sequencing. So you may know that the sequencing of the virus is done by amplification sequencing overlapping amplicons. So the problem with viruses, so usually when we want to sequence a bacteria, so first we culture, we grow the bacteria, and we sequence actually and isolate of the bacteria. So we have almost pure bacterial DNA to sequence, which is kind of not easy, but it's easier. But for viruses, it's not the same thing, because we cannot culture the virus. So we have to start from the clinical specimen. And this is something which is difficult. So you can either do what we call metagenomics, so sequence everything that you have in your sample. But if you sequence everything that you have in your sample, so the virus will represent only, let's say, less than 1% of everything that you will sequence. So it's very expensive. So we have to be smarter than that for viruses. And what has been designed since the beginning of the sequencing of the virus is a set of primers that amplifies overlapping regions of the virus in clinical specimens. So we have around 100 of those amplicons that we sequence, so 2 pools of amplicons. In Quebec, our capacity, so these primer set were designed by the Arctic network here. In Quebec, we have a capacity of 1,800 samples per week, and we use both Illumina and Nanopore for sequencing. So for the Illumina, the pipeline that we use, so this is a pipeline that we use for Illumina reads. So this pipeline was developed by the team of Guillaume Book in McGill, by the Bioinformatics team. And if you want to see, to have more information about the detail of the pipeline, you can have information on that link. So basically, it's the main steps are host removal, because you still have some human reads in the viral reads. So it's easier to if you remove those reads, and it's also for privacy concern, because when we publish those the raw reads should not have human reads in them. So that's why we do that step. We trim the sequences. Then there is an alignment on the reference genome. We do the variant calling to identify mutations, and then there is the consensus sequence, which is assembled. And finally, the plots and metrics are produced. So this is something that I usually, this is a slide that I put usually for people which are not, for an audience which is not used to genomics, because it's not clear usually for people what the resources which are needed for sequencing SARS-CoV-2. And usually people think of that as a black box, where you put the clinical specimen at the beginning and then automatically you have the sequence at the end, and that's it. And so, and I don't think that it's a problem with that specific audience, but in Quebec, for the genomics part, it takes like 2 days for the library prep, around 24 hours for sequencing, at least with Illumina, and it costs something like between $75 to $100 per sample. There is, of course, automation with liquid handlers, and only the technician with the most skilled in molecular biology do those kind of library prep. For the bioinformatics part, a classical run, it's 8 hours computing on the Compute Canada server, 80 processors, and each run is 1.5 terabytes of data. Of course, we have an automated pipeline, and once again, it requires important bioinformatics skills for that aspect. So it's not that easy. I mean, it's a quite complex structure. So the challenges that we faced with that project. So the first and the main challenge that we have are delay. And it's mainly delayed due to logistic constraints. And the problem that we have in Quebec, so that's mainly the fact that we use a biobank, and that the biobank is far from hospitals. So mainly, most of the hospitals are in Montreal or Montreal area, and the biobank is far away in the north, in Chicoutimi. So the samples are sent to the biobank, aliquot in the biobank, and then back in Montreal for sequencing. So we lost a few days just because the biobank is far away. So it was easier at the beginning of the project because we didn't have the resources to receive all the samples, but at the end, now we have a long turnaround time because of this logistic aspect. So more than 2 weeks for the sequencing of samples, and the hospitals that don't send their samples every day. So we have a delay also between the collection date and the date where samples are sent to the biobank. So it's even more long. We have also delay for the data integration and the reporting. And something which is really crucial in bioinformatics during the flow of the data is the format. So we spend our time doing script to reformat data, to reprocess data in one format, another. So it's the main part of our work, I would say. And the fact that LIMS laboratory management system are not harmonized between the labs, and we all work with different systems, different databases. So it makes the whole system very complicated. And so I would say that one of our weaknesses is this aspect, data integration and reporting. The sample tracing, so when I'm looking for a specific sample for an outbreak analysis, for example, so the sample could be in the hospital, on its way to the biobank, on its way to the sequencing centers, and if I need the information about that patient, I should contact public health authorities to have information from their database. So overall, my sample is somewhere in there, but sometimes it's really difficult to know exactly where is my sample and all the information about the sample. So the sample tracing is something which is complicated and which was difficult for outbreak investigation. Other aspect is that the LSPQ, the public health lab, has not been designed as an high throughput lab. So we are a reference lab, so we have limited resources. So it makes things a bit more complicated, and we have a recurrent shortage of highly qualified personnel, which is another something which is a recurrent problem in the lab. So our objective going forward is to improve lab workflow, the sample management and the tracing, to improve data workflow and to implement reporting tools. So this is the main objective. But we had great successes, I guess, I think. And one of these successes in my, which is important for me, is the strong genomic network with sequencing center partners. And this is something that will last even after the pandemic. So this is great to have this huge and this genomic network between sequencing center and public health lab. And I hope that it will help for other projects. We have also good collaboration with academic researchers and clinical laboratories. So of course we could always do more research, and it's always what I want, but we have limited resources, and the collaboration that we have built I think are great. Another success, I would say, is the close and fruitful relationship that we have with epidemiologists. So we have committees with people from public health, so APs, and from the lab, so me and my colleague. And we have also, in those committees, we have also researchers. So having everybody around the same table is something which is really great for the project, because everybody is going in the same direction. And so when we need information, it's easier to have information when they are included in the same meeting. So for the research collaboration network, so for phylogenetic analysis, we have this great collaboration with the team of Jessie Chaperoux and Julie Hussin, which are now also part of Covarnet. So you have the link for those projects. We have this fantastic functional analysis group, the Sentinel COVID Quebec, that I will, maybe if I have time, I will tell a little bit more about that project. We have also collaboration, I'm part of the CanMod project, which is Canadian modeling project, and we have a project with Finlay Maguire and Samira Mubarak. We want to compare the interprovincial dynamics within vulnerable population, so between Ontario and Quebec. And of course, we have projects with the epidemiologist in LSPQ. And also this link is something which is not really a collaboration, but what we try to do is so the BQC19 is a biobank of human samples. So it's not the nasal swabs. So it's blood or so human samples for patients who had COVID-19. So this BQC19 is collecting samples, and what we do in LSPQ is to ensure that we will have the virus sample for the patients which are enrolled in that cohort, so that we will have both the virus and the host information. So yeah. So I will tell you a bit more about the outbreak analysis and this article about the early introductions of the SARS-CoV-2 in Quebec. And if I have time, a bit more about that project, about the Omicron spike recognition with the functional analysis group. So for
I guess I have a question at this point. It's about like when this whole network started getting on its feet. Like, was there an expansion at the at the sequencing facility that are existing in Quebec, or was it mainly just a reorientation of sequencing resources that were used for other purposes for the emergency of COVID-19?
Yeah, so they were both, and I don't know if you remember, but at the beginning of the pandemic, everything was on hold. So we couldn't do anything except sequencing SARS-CoV-2, and actually all the research projects were stopped. because of that. So we had these resources which were available in sequencing centers. So at the beginning of the pandemic, it was just using those resources that were available for sequencing. But then after that, there was another aspect, which was what we call the capacity building, and it was increasing the capacity of, so it was mainly public health labs, so more resources, but specifically for the surveillance of SARS-CoV-2. So first phase was using the resources which were available because they didn't have anything else to do, and second step was to add more resources.
And how much in this like process do you think that like different provinces in Canada like developed their own surveillance network independently, and how much of it was like seeing what others did? Because like last year, for example, we had a talk by Dr. Natalie, and she told us a bit about like BC's own COVID surveillance network, and I'm now like trying to like put these together in my head and like, oh, like how much learning was there between Quebec and BC and other provinces?
Well, actually, it was, we had weekly meetings together. So Natalie is also part of the Concagene network, as all other provincial public health laboratories. So this is really fantastic because all people from all public health labs were together in the same meetings. But for sure, we have differences. We have differences because we don't have the same equipment, we don't have the same constraints. So of course we implemented our own version of the surveillance network because of these constraints. But because we were all working together, we had quite an harmonization, I would say, of what we did.
Cool. Okay, thank you.
So the outbreak investigation, I will tell you about 2 requests that we had for outbreak investigation in long-term care facilities. And these outbreaks happened shortly after the beginning of the vaccination in Quebec, which was the beginning of 2021. So the 2 long-term care facilities, I will call them A and B. It's CHSLD, CHSLD in French. You will see in my slides. And so there were, it was huge outbreaks, and there were cases amongst vaccinated patients. So the question that we had from people was, is the vaccination working actually? Why do we have cases among vaccinated patients? And is it due to a variant of concern? So variants which have which are more transmissible or more severe compared to other SARS-CoV-2 variants. And we use genomic epidemiology to allow the reconstruction of the transmission chain. So I guess that people in that audience are quite familiar with this kind of analysis, but just to briefly summarize. So the virus spreads by rapidly replicating many times within its host, and the transmission to a new host repeats the process and an epidemic begins. And the rapid and extensive replication of a virus genome results in copying mistakes. These random mutations are almost always minor and inconsequential, but can be identified and exploited to track the epidemic. And so with the sequencing of the virus, so here we have a mutation in dark blue, and one in light blue. So with the sequencing of the virus, the collection date and the place, the transmission chain can be reconstructed. I would say that the limit of this technique is the fact that the virus so there is one mutation every 10 to 15 days. So if you have transmission between inside those in less than 10 days, you will not see differences in the sequence of the virus. So from this person to this person, it will be the same sequence. So this is the blind spot of the technique. So genomic epidemiology can track the spread of the virus, so locally for an outbreak or globally for international transmission. So first, so we sequence, of course, the samples for that investigation, and for the CHSLD-A, we have 42 cases which were submitted. CHSLD-B, 170 cases. So the percentage of missing sequences is a proxy for the quality of the sequence. So you can have long stretch of the genome which are not correctly sequenced. So it's another blind spot. So you cannot say what is going on in that region of the genome. So usually for 70 to 80% of the genome, the quality is good enough to do this analysis. In the case of CHSLD-B, we had environmental samples that were submitted, but the quality was low, so it was not included in the analysis. Then we do a phylogenetic reconstruction to find the number of introduction in the facilities, and for that we use the next range tool, which is really fantastic also. So we use it a lot in the lab. So there are two parts in that tool. So the augur and the auspice part. The augur is this part which is the pipeline, the bioinformatics pipeline that filters, align, do the tree, refine the tree, and finally export a JSON file. For the visualization with auspice, and you have this nice phylogenetic tree and the transmission on the map. So it's really a fantastic tool, and you can use, of course, metadata about your sample that will be mapped onto this phylogenetic tree. So this is really useful. So in our case, we have for CHSLD-A a single introduction here. With a variant which is B11157. Nobody knows what it is. Everybody has it was not a very important variant actually. And for CHSLD-B, 7 introductions with 4 major clusters. So a really different picture if you compare those two outbreaks. So for CHSLD-A, we have a single cluster and the single lineage. And something which is confusing sometimes for epidemiologists is that they think that if there is a single lineage, that means that there is a single introduction. But you can have more than one introduction with the same lineage, so it's important to keep that in mind. In our case, it was a nice cluster and the single introduction. So a phylogenetic analysis is required to identify the number of clusters, not just only the lineage information. And it's important in your phylogenetic analysis to include enough sequences in the context, because if you don't include enough sequences, or your sequence might just group together, but just because there is not enough context to split the cluster. In the CHSLD-B, we have some sporadic cases. So for the interpretation of that, it's important to keep in mind that not all the cases were sequenced. So it might seem sporadic cases because we have only one sequence, but actually if we add all the sequences of that outbreak, maybe we would have more. Then we do the epidemic curve to visualize the dynamic of the introduction. So in the CHSLD-A, a single lineage spread in the facility, and in the CHSLD-B, we have different introduction during the outbreak investigation. So something which sometimes is difficult is to find the information of the symptoms onset. So usually we don't use this information because we don't have the date of symptoms onset. We only have the collection date. But when there is an investigation, everybody usually is, we have a campaign of collection. So you end up with many specimens having the same collection date, which is more difficult to interpret. It's important to have genomic information and epidemiological information, because genomic information alone is not enough to interpret the situation. So, for example, if we joined the information, the epidemiological information to the genomic information in that case, and we saw that the introductions were mainly by health workers, and also with the epidemiological information and investigation, we saw that genomic clusters were grouped by floor, something that you can't see, of course, with the genomic information only. So it's important to work together with epidemiologists for the interpretation of what is going on in an outbreak. So then we identify sequence type, so what we call sequence type. We define sequence type are sequence that have less than 4 different mutations. So that's an arbitrary threshold that we put. And usually those sequence types, so A, B, C, D in that case, correspond to the cluster. Sorry for the French, I didn't translate it. So then we reconstruct the transmission chain. And something which was interested in that case, which was quite a complicated case, so the CHSLE-B, is that we realized that there was a sample which was submitted for a reinfection, and we realized that it grouped together with this cluster, the B cluster, and that it might be the source of the introduction in that cluster. So a sample which was submitted and sequenced not at all for the purpose of this outbreak investigation ended up being kind of key in the interpretation of the situation. So that's great also to have a random sequencing of all the cases in the population, because you have a far better picture of what is going on. So in that case, it was funny because it was reinfection, and at the beginning we had lots of questions about whether it could be possible to be reinfected and the time between first and second episode, etc. So it was interesting. So in conclusion, for the outbreak analysis, so in CHSLE-A, the phylogenetic analysis identified a single source for this outbreak. And in the B CHSLD, so it was 7 introductions. No VOC was identified, variant of concern was identified in the CHSLD-B, so it's not the cause of this large outbreak, and a variant under investigation in Quebec, the lineage B1160, was identified in that case, but it was only a few cases of that outbreak which had that lineage. So in that case, the spread of the virus in the long-term care facility is caused by difficulties to control the transmission of the virus, a unique introduction, many cases. And in that case, the spread of the virus in the CHSLD is caused by difficulties to control introductions. So many introduction, fewer transmission. So it was interesting to see this different picture. And the exceptional characteristic of this outbreak, the high number of cases and the secondary attack rate, with cases infected more than 40 days after vaccination, could not be attributed to lineage with exceptional transmissibility or resistance to vaccination. It was really classical SARS-CoV-2 variant. So yeah, not variant of concern in that case. So I guess that so it's it should be the end of the talk. No,
we still have about 10 to 15 minutes to go.
Okay, so I will just finish with that part, which is the analysis that I prefer, so that's great, and my talk. Okay, so the second analysis is the analysis of the introductions of SARS-CoV-2, but at the beginning, really at the beginning of the pandemic in Quebec. And this is a collaboration with Jessie Shapiro in McGill, and the main author is Carmen, which is now working for the National Microbiology Lab. So fantastic work. It's published in Genome Medicine. So the questions was around the introductions during the first wave. So it's really at the beginning of the pandemic. And the study is between the 25th of February until the 1st of June. So the 25th of February, so why this date? It's because it's the first case in Quebec. So the first SARS-CoV-2 case in Quebec is the collection date is the 25th of February, and 1st of June, it's because it's more or less the end of the first wave in Quebec. So during that period, we collected close to 3,000 sequences of high quality. So it represented around a bit less than 6% of reported cases during that period. We had 45,000 cases during that period in Quebec. So if you look at this graphic, you have the sequences in blue, and in gray the daily case count. And I also put the important dates also on that slide. So the spring break in Quebec was between the 29th of February and the 9th of March. So here, so it will be important for the rest of the analysis. And on the 16th of March, it was the border closing and the lockdown. And with the 3,000 Quebec sequences, we also used more than 12,000 international sequences for our analysis. And so the first analysis that we did, so we put all the sequences in a phylogenetic tree, and we did a phylogenetic analysis, of course, and ancestral state reconstruction, ASR, to identify the non-Quebec to Quebec transition nodes in the phylogeny, and so to find where does those sequences came from. And actually, we identified more than 500 introductions by this analysis, and what we saw is that by the phylogenetic and the ancestral state reconstruction, that those sequences originate from Europe mainly, with 34% of the sequences from Europe. And we had only a few sequences from Asia. And so you must remember that, so the first case in Quebec was the 25th of February. First sequences in China were back in November, December. So what we detect with the phylogenetic analysis is the intermediates. So no, not the original sequences, the original sequence, of course. And so we had information from the phylogenetic analysis of the origin of the sequences, and we have also collected the travel history of the patients. And what we noticed is that there is a difference between the travel history. So we don't have the travel history for all the cases, but the proportion is different between the travel history and the phylogeny. And in particular, the Caribbean and the Latin America, there is a quite important difference. And this is something that we found quite funny, is that actually you don't bring the souvenir that you think. And what we realized is that during the investigation, so people told the public health authorities that they went in vacation to the Caribbean, but actually in the Caribbean, they had contact with people coming from all around the world, and they don't bring back sequences from the Caribbean, but they bring back sequences from Italy, from Netherlands, etc., etc. So actually, the epidemiological investigation was less accurate, I would say, than the phylogenetic analysis, for which we had the correct origin of the sequence. And this is something which is very interesting because it highlights what phylogenetic analysis can bring to this kind of investigation. So DNA doesn't lie, so it tells you the truth, so the exact origin of the sequence. And so in that case, the Caribbean has been a global hub for virus exchange during the spring break. And then with the analysis of the time to the most common ancestor, we tried to date the likely time of introduction of the virus. And this analysis was categorized by the origin. So you have the USA here, Latin American and Caribbean, Europe, Canada, Asia, and Africa. And what you can see, so this is the time. This is a spring break here in gray. And you can see that before spring break, you had only 12 to 16% of the introduction, but the majority of the introduction were after spring break. So 78 to 82% of the introductions occurred after spring break. And something which is highlighted there is that you can see that there is a peak of cases, and this peak of cases is actually due to the snowbirds, who were returning to Canada after the border closure here, and you can clearly see that peak. So what we found is that the majority of introductions occurred after spring break, but then we asked the question. Okay, there were these introductions, but were these introductions successful? And we realized that most of the successful transmission were the earliest, the most successful transmission were occurring actually during spring break. And among the more than 500 introductions that we have identified, 60% were singletons, so only one single sequence. 90% were in cluster with less than 10 cases, and 5 to 10, so less than 2% caused more than 50 cases. So, and those big cluster are highlighted here. And the 10 introductions leading to more than 50 cases occurred during spring break and led to a total of almost 1,500 cases, 48% in this study, so the majority of introduction. And these lineages came mainly from Europe. So what was surprising is that because most of the introduction occurred after spring break, we thought that it was the main cause of the transmission that we observed afterwards, but actually it's the minority of cases that led to the majority of transmission. And the other introduction have been actually quite well controlled. So we had few super spreading events that gave rise to most cases. So you can control most of the cases, but if it's the only little one that you won't control that will lead to the majority of cases in the pandemic. So in summary, we have identified more than 500 introductions in Quebec during the first wave. a minority of introduction, 5 to 10 during spring break, gave rise to the majority of cases, and they came mainly from Europe. And it's actually, we are not the only place to observe that. This scenario with super spreading events is similar to what happened in other countries, so UK and Massachusetts. And the majority of introduction happened after spring break because of the repatriation period, but did not lead to lots of transmission. So we can say that it seems that quarantines, isolation, and countermeasure were quite efficient. And the imported lineages originated mainly from Europe, and it's similar to what was observed in other cities in the East Coast, but it's different from what was observed in the West Coast with lineages more from Asia. And with that, I will just skip the rest of my talk and just thank all the people who participated in these studies. Thank you.
Well, thank you so much. That was really, really informative and quite pleasant to attend. I do have a question, I guess I'll start with, and if anybody has questions, please post them. Or again, raise your hand if you want to ask the question yourself. My question was about the earlier cases with the long-term care settings, the two cases that you discussed, A and B. I guess the question is like two parts. The first one is, how long did that take from like the first case that was identified in like one of these long-term settings to them requesting that you investigate this, and then the turnaround after that for getting an idea that, oh, this is one lineage here, there's multiple lineages here. And the second part, which is more like perspective, is like, I guess, how much of that time was because it was the first time that you're like earlier developing the protocol for how to do this, and if you, I guess, want to do it again, how much faster could that potentially become?
That's a very good question, actually, for public health. So mainly, it's the question is outbreak investigation with genomics actionable for a public health intervention? So can we use that to do something for this outbreak? And the answer is quite disappointed. But it took too much, it takes, at least in Quebec, too much time from the time where we collect the specimen, the time that the specimen, we find the specimen. So I told you it's very difficult to know where are my samples. So it's really, it takes weeks sometimes for some of the specimen. And then after that, sequencing is the easy part. In that investigation, when I have all the samples in the lab, so it's quite easy because I just put them in the sequencer. Then the bioinformatic pipeline is automated, so it's not a problem. And then the analysis. So the only things that takes time is having people available. So me. To do the analysis. So finding time to do the analysis, but. Really the major problem is finding the samples. And so as for now in Quebec, it's something that you can do to analyze what happened, but it's not an analysis that you can do to, it's not something which is actionable. So it's not something for which that you can wait for to take a decision. It's too long. It takes too long. And maybe the future will be to have a point of care sequencing equipment and everything in the laptop to do the analysis. So maybe it's the future. But as for now, it's really more useful retrospectively.
do you know of any locations like around the globe that maybe is not there yet, but is like pioneering or like trying to push the limits of what can be actionable for such surveillance?
Well, I've heard in New Zealand that they are using, for example, so they are using nanopore for outbreak investigation and fast sequencing and analysis. And they were, so I don't know the situation right now in New Zealand, but at one point they had quite 0 case at all of SARS-CoV-2. And so they had this fantastic fast response team and they could have access to the samples immediately, do the analysis right now, and have the answer in few days. So that's, I mean, if you are under 1 week, that makes things actionable, actually. So there are places. So technically, that's possible, but it's only a question of having all the logistic aspects solved and everything in place and available and the resources available. So it's possible.
We have a question from Angela. So thanks for the nice summary, Dr. Sandrine. And I was wondering, to what extent does higher sequencing representation earlier in the study period affects the conclusion that introductions prior to spring break caused the majority of sampled cases? Would you expect the same conclusion if all infections were sequenced?
Yeah, another excellent question. So the first analysis that we did, for the analysis that I've showed, we used a bit less than 3,000 Quebec samples. And we did a first primary analysis with 800 samples. And with 800 samples, we found half introduction, so 250. So for sure, the more you add samples, the more introduction you could detect. So is it representative? I don't know. So this is, of course, a limit of the method. So we can say that we had at least 500 introductions, but we may have more. But would we have a different conclusion to the fact that the majority of introduction occurred after the spring break? I don't think so. I think that it's relatively in line with the epidemiological investigation that were done at the same time. And the fact that a minority of introduction led to the majority of transmission. So because it has been also observed in other places, so this is something which is consistent with what could be expected. So I think that it's representative, but I agree with your comment on the fact that, of course, if we could sequence everything, we would detect more introductions.
Cool. Thank you so much. I wanted to ask you about like the challenges of the specifics of the sequencing platform, whether like you mentioned nanopore, and like nanopore comes with this promise that you can do much more rapid sequencing and potentially even like with a minion on the spot. But how much does that like push some of these some of the challenges downstream, especially for like the analysis pipeline, because of the higher sequencing error rates, the higher noise that you could have?
Well, for nanopore, so I've seen that there is a John on the line which is doing sequencing, and he is quite an expert on nanopore sequencing in BC. But I don't know if you want to talk about the higher error rate of nanopore, but this is not really an issue for SARS-CoV-2, because I mean, there is usually we sequence up at something like 5,000x or 10,000x. So at this coverage, I mean, this is the 10 or 15% error rate. This is not a problem at all. So for samples for which we have lower viral load, that could be a problem. But for the other sample, the coverage is so high that it's not a problem. And the best performance, of course, that I've seen in terms of turnaround time is with, of course, nanopore. So this is something, a technology for the future. So for a clinical investigation and fast answer, so using this point of care tools like MinION for nanopore, that I'm sure it will be the future of clinical investigations. I'm sure, John, you agree with me.
Yeah, so I'll just quickly say, I mean, effectively, what we've done here in BC is also, our major production is effectively on the Illumina platform, and obviously we have that higher throughput. So we're kind of doing, I guess, consistently around close to 4,000 sequences a week. What we did do with when Omicron initially arrived, we did quickly shift to like a rapid nanopore, and the gain there was, it was just the kind of, we gained probably about 24 hours on, from, because the sequencing, depending on the coverage, as Sandrine said, you only have to sequence for a limited amount of time, and then you can analyze on the fly. So we were using some sort of live, like real-time integrated kind of analysis platforms where you'll go and sequence until we were effectively getting, say, when we hit 20x on 95% of the genome, we'll make a call. And so you can call things, the first one we did, we probably called, we did sequencing for probably half an hour. And then the analysis was done in a few minutes after that, using some of the systems from Matt Luce and stuff from the UK, has his Minotaur sort of thing. But I think, as Sandrine's kind of alluded to, actually, the actual sequencing time, to be perfectly honest with you, is actually not that significant sometimes, depending on the logistics up front, right? I mean, we've in BC, we're in a luxurious position, actually, the way it's set up, actually, that, we don't, I guess the plus is we don't have that biobank thing going on, but then I guess the disadvantage is then you have the biobank and we don't, right? Because, we've been in a situation where, we've had a lot of samples and we're at the point now where we can't keep everything, so we sequence things and then we'll end up keeping a certain fraction of those. Like, we've just gone through and selected, for example, I went through and looked for, anything that we've had, say, 20 cases of in terms of a unique genotype, we'll keep representative samples of that just in case we want to go back, at some point and do anything with that. But generally, when we've sequenced something, and then that's it, the sample, we'll keep it for a while in case we get requests for outbreak investigations from some of the health authorities and those sort of things. That's the only reason we keep them, in case there's a retrospective call to say, can we look in a little bit more detail on this, sort of outbreak in one of our hospitals or something like that. That would be the reason for that. Because when we got to a point where we weren't sequencing everything, obviously then we are not in a position to then, we don't know which samples they want to have, so we have to keep them for a certain amount of time. So it's all about, what's required, and if you like, the public health, demand up front, and how we're trying to help that with the outbreaks and, genotypes and things like that. But I don't want to go on too long. But yeah, no, like I say, there's all sorts of little things, like Sandrine said, about, you can speed it up a little bit in some of the capacity things, with MinION, with GridION, and those sorts of things. But also, we did a lot of work on, really speeding up the Illumina pipelines as well. We just put out one where we're doing a lot of cut down protocols, and then also saving a lot of plasticware, because this was a big concern. Originally the protocols that were being used were using like 34 boxes of tips. We cut that down to 4 boxes of tips for a library, right? Just by hacking and getting, changing the protocols and, using some automation. And what people have to also realize is, it's all right. It's easy to say we're going to automate it all and we're going to get it, but then you have to work within the window of what plasticware can you get, what robot could we get, what what supplies could we get. So we ended up going with we use like Epmotions, which they're kind of like, I guess, a mid-range robot. They're not the one that high-end genome centers would choose to use, but they were the ones that, one, you could actually get the instrument, you could actually get the plasticware for, and so we were waiting for months or a year for Hamilton stuff, or we just couldn't get supplies. So it was more a case of, we ended up, going a route that was just what would work for us. And each jurisdiction was in a different position like that. I mean, so it was a case of how could we most expeditiously move to actually provide what we needed to provide in the time. And that was kind of, and so we look at all these different platforms and stuff. And obviously, we do an amalgamation of, whichever one kind of works. But we've kind of gone back now because it's more of a surveillance and monitoring, except for little outbreaks. Because obviously now we were always trying to do as much sequencing as we could to maintain a throughput, and we tried to be consistent and not go up and down too much, just for resources with personnel as well, because we don't want to it's all right saying do this, but we have to keep people, and they have to be we have to be able to work and consistently work. We don't want to like break people by suddenly saying, you know what, today we're going to do 100% of everything, and we have to work 24/7 to do that. It's like, so we ended up fixing the capacity, and then if we caught 50% of cases, and then like you hit a wave and you hit 10% of cases, you just have to accept that. And we chose to just try and be consistent and maintain throughput rather than chase things too hard, after a while. Anyway, sorry, that's me going on enough. Sorry.
Thanks, John. I guess maybe I'll finish with a question about again, like a bit of the outlook of this beyond the pandemic and beyond COVID-19. Like, how much do you think that the urgency of COVID-19 made this, demonstrated this as an actual example that public health can see, oh, okay, we can actually use genomic sequencing? And how much of it, do you think that public health would start thinking for this also, beyond COVID-19, for infectious diseases, and I don't know, potentially even non-infectious diseases, but mainly for infectious diseases like the flu, with how much is the case sold, I guess?
Yeah, so this is, I think, something which is really important for the future of public health labs, because if we take the example of Quebec, for example, so before the COVID-19 pandemic, so we had only one validated clinical microbiology test based on genomics, so only one before COVID-19. And this is something which was a bit surprising for me when I arrived at the Public Health Lab, because we have been sequencing bacterial genomes for years. We know how to analyze, annotate, assemble everything, search for resistant genes. We know how to do phylogenetic analysis, and we do that for years in academic labs. So why don't we use all these resources in clinical labs? And I think what this pandemic has changed actually is the fact that people realized that actually it could be useful. It's not something which is just for academics and only for research. We can use genomics for to answer public health questions. But and we had this great capacity building component for Concogene, for example, so the Canadian project, which gave money for equipment and resources in public health labs. So the resources are there, the new equipment, the sequencers. So there is a new area, I think, that will start with all these resources, and people will start thinking, well, we can consider that really useful for public health. But I think that the road is still long from having a research project on the assembly and the analysis and the annotation of the bacteria, and the fact that it's useful for public health. And so I think that we should work to fill that gap. So to implement validated protocols, to implement a more stable bioinformatics pipeline, to train people in the labs, to train them more. So there are different aspects that we can work on, but for sure it will be, we will have the COVID-19 pandemic is a transition for sure. So things will change. So I think that we will have maybe more opportunities for bioinformaticians to join public health labs in the future, hopefully.
Hopefully. Well, with that, Thank you so much for the great talk, and thank you, Armita, as well for your introductory talk, and the audience for attending. And hopefully we'll get the YouTube with a high turnaround uploaded, so we can share it. And thank you so much for everything. Have a good day,
Thank you.
everybody. Bye.